Hello everyone, welcome to AIMS Biology. Previously we have discussed about the discovery of nucleic acid or DNA and some basic things about DNA, nucleotide, nucleoside and polynucleotide chain. Today let's discuss about the structure of polynucleotide chain clearly. Okay, we have studied that the basic unit of DNA or nucleic acid is nucleotide. Okay. We have also discussed that nucleotide consists of a pentose sugar, a nitrogenous base and a phosphate group. So, let's discuss it clearly. The basic unit of DNA is nucleotide. Okay. Which has three components, which we have already discussed: a pentose sugar, a nitrogenous base and a phosphate group. Okay. गोटे न्यूक्लियोटाइड तीनटा जिनस को लेकी गठित, गोटे पेंटोज सुगर, गोटे नाइट्रोजनस बेस एंड फॉस्फेट ग्रुप. If we remove phosphate group here, a pentose sugar and nitrogenous base together form a nucleoside, which we have already discussed. Okay. So let's see how they are attached together to form a nucleotide. Okay. जदि आमे गोटे फूल माଳ तैयारी करिया को चाहिबा हमको कोन कोन दरकार, किछि फूल दरकार, सूता दरकार, आ कोन दरकार, गोटे छुंची दरकार. तो आमे कोन करबा, फूल रे छुंची साहाय्य रे फूल को आमे पिन करबा एवं ता थ्रेड सूता सहित गोटे गोटे फूल जोड़िबा. तो फूल जोड़िले गोटे माळ तैयारी हबो. जदि आमे फूल, सूता आ छुंची सब जाको गोटे जगह लेके रखी दबा तो सेटा गोटे माळ हबो कि हबोनी. से भलिया जदि आमे पेंटोज सुगर, नाइट्रोजनस बेस एंड फॉस्फेट ग्रुप को एमती छाडी दबा तो सेटा गोटे न्यूक्लियोटाइड फॉर्म करिवो नाही. तो ताको आमे कोन करिया पड़िवो, जोड़िया को पड़िवो. हैला. तो केमिति गोटे न्यूक्लियोटाइड फॉर्म होछि, लेट्स सी. A polynucleotide chain shows different linkage or bond. Here one pentose sugar, here another pentose sugar. Okay, this is deoxyribose sugar and this is ribose sugar. Deoxyribose sugar present in case of DNA and ribose sugar present in case of RNA. Both are nucleic acid. This is deoxyribonucleic acid and it is ribonucleic acid. Both are nucleic acid. Okay. So a pentose sugar. First name नहीं गले उटे pentose sugar नहीं गले. Then a nitrogenous base. Then we have to add here one nitrogenous base. Where we add the nitrogenous base. Okay. Let's see. A nitrogen base is linked to the pentose sugar at first carbon atom. Okay. First carbon. गोटे नाइट्रोजन बेस को हमें ऐड करिबा कोठी फर्स्ट कार्बन रे ऐड करिबा. ओके. तो एटा फर्स्ट कार्बन बा एक नंबर पर कार्बन बोली हमें केमिति जानिबा. ऑलवेज just write to the oxygen atom with the first carbon. Okay. Always just write to the oxygen atom. This is oxygen and just write here the carbon atom present here is the just write to the oxygen atom. Here we can add the nitrogen base. Okay. Nitrogen base. Then how we add the nitrogen base? Okay. N-glycosidic linkage. A nitrogen base is linked to the pentose sugar at first carbon atom through N-glycosidic linkage. ए जो लिंक बा बॉन्ड क्रिएट होला जहा द्वारा गोटे नाइट्रोजन बेस आड होला कहा सहित पेंटोज सुगर सहित तो एटा को आमे कहले कोन कहले ना एन ग्लाइकोसाइडिक linkage. The nitrogen base is added to the sugar, pentose sugar, with a linkage that is known as N-glycosidic linkage or N-glycosidic bond. Okay, link or bond. A nitrogen base can added to a pentose sugar through N-glycosidic linkage. Okay. Then come to the phosphate group. How and where a phosphate group will be added to a pentose sugar to form a nucleotide? The phosphate group will add to the 5 dash. It is also known as 5 dash prime, 5 carbon atom or 5 dash prime. This is also 1 dash, 2 dash, 3 dash. Okay, or 3 dash or 3 prime, 5 dash or 5 prime. Here the phosphate group is added. Okay. After addition of 3 things, this will be called as nucleotide. Okay. A nucleotide consists of a pentose sugar, a nitrogenous base and a phosphate group. Okay. And a nitrogenous base added to the pentose sugar with or through a N-glycosidic linkage or N-glycosidic bond. Okay. And the nitrogenous base always added to the first carbon atom. Okay. Next, phosphate group is added to the 5 dash or 5 carbon atom. Okay. And the phosphate group is added through a linkage known as phosphoester bond. Phosphoester bond. Okay. Now a nucleotide is complete. Okay, let's see how a dinucleotide will form. Suppose this is another nucleotide. Okay, 1, 2, 3, 4 and 5. Here also present one phosphate group and here present another nitrogen base. Okay. Then how these 2 nucleotide will attach? 2 nucleotide will link to through the 3 dash to 5 dash through phosphodiester linkage. Okay. This is one phosphate group is present. If we add it to the hydroxyl group of first nucleotide, then it will form a dinucleotide. Okay, dinucleotide. Di means 2. Okay, and 2 nucleotides are attached together through phosphoester bond. Here is 2 phosphoester bond, one with 3 dash end of first nucleotide and second with the 5 dash end of second nucleotide. So a dinucleotide form through phosphodiester bond. Okay, दीटा phosphoester bond एठी दीटा nucleotide को attach करूछंती सेति पई कोन हला the nucleotides or dinucleotide formed by phosphodiester bond. Likewise, this a polynucleotide chain will form. Let's see. Suppose this is another nucleotide. Here nitrogen base present in first carbon atom and this is the second carbon, here hydrogen group present. This is the 3 dash or third carbon atom. Here one hydroxyl group present, and this is 4 and this is 5. Okay. Here another phosphoester bond will form with the second nucleotide to the third nucleotide. Okay. So likewise this many nucleotides are attached together through phosphodiester bond. Okay. तो न्यूक्लियोटाइड गुड़ा को परस्पर सहित केमिति जॉइंट होउचंती? They are joined together through phosphodiester bond. Okay, this is very important question, that is how the nucleotides are attached together, or through which bond the nucleotides are attached together. The nucleotides are attached together through phosphodiester bond. Okay, so like this many nucleotides can attach to form a polynucleotide chain. Okay, like this in case of RNA also same. The backbone in a polynucleotide chain is formed due to sugar and phosphate. Okay, गोटाए न्यूक्लियोटाइड रे सुगर आ फॉस्फेट दी जण मिसिकी कोन करूचंती? बैकबोन तयारी करूचंती. बुझियला? Morris Wilkins and Rosalind Franklin obtained the X-ray crystallography of DNA. Franklin and Wilkins. Wilkins and Franklin obtained the X-ray crystallography of DNA. The DNA रो X-ray टाकू के बार करथिले? Wilkins and Franklin. Okay. Irwin Sargoff put forwarded certain rules for double standard DNA. Sargoff कौन करथिले? ना किछि रूल प्रोवाइड करथिले डबल स्टैंडर्ड डीएनए पाई. तो डीएनए may be single standard or double standard. It is vary from organism to organism. Let's see some example. Organism, genetic material, number of nucleotides. DNA is the genetic material in almost all organisms except some virus. Number of nucleotide. Number of nucleotide can define the length of DNA, which we have studied in the previous class. Let's see. Organism Psi-174 bacteriophage contain single standard DNA. SS. S stand for single. S stand for strand. ssDNA, single standard linear DNA. Linear, like this. Linear DNA. And the length or the number of nucleotides are 5386 bases. Only bases because it is single standard. Only bases. Okay. Next, lambda phage contain double standard DNA which is linear, and it contains 48,502 base pair. Base pair because it is double standard DNA. गोटे बेस आ गोटे बेस दुटा बेस कोन हेचंदी पेयरिंग हेचंदी डबल स्टैंडर्ड. ओके. E. coli contains double standard DNA which is circular. Double strand but circular. Okay. This is double standard and circular DNA present in E. coli. Okay. In human, double standard DNA which is linear and number of nucleotide is 3.3 into 10 to the power 9 base pair which is haploid. Okay. So this is all about the structure of polynucleotide chain and some discoveries related to structure and composition of DNA. In the next class we will discuss about the structure of DNA, that is double helical structure proposed by Watson and Crick, and the Chargaff's rules. Till then, keep watching and keep learning. Don't forget to subscribe my channel and follow me on Facebook for more videos. Thank you.